Right, welcome to your video for lecture 21. So before we get started, since this is the second last lecture video before you're off to study for your finals, I just wanted to give you a few more pointers for how to maybe approach your final exam studying by mixing together some of our older tips as well as some new ones as well. So first thing you definitely want to do as soon as possible is set up your study schedule, as we talked about early in the semester. So remember to work backwards from your last exam and then work your way back just to see how you're going to divide up the time for studying for each of your exams and make sure you have enough time for each one. Now, once you've kind of decided how many days you have to study for each exam, then a new tip for you is to find a way to set the right pace for that studying. So for example, if you find that you only have 5 days to prepare for BIS 202, for example, maybe you'll set 2 of those 5 days for reviewing the stuff that came before midterm 2, the stuff that was covered in midterm 1 and 2. Maybe you'll spend 3 days reviewing the newer stuff. Now, once you set up the number of days you're spending on each set of topics, for example, you can take a look and see how many kind of weeks of lecture are you going to have to then cover per day. Like at what pace do you need to study to be able to finish within 5 days? So with this particular division, you'll be trying to cover 4 weeks of lecture per day, and then for the new stuff, 2 weeks of lecture per day, right? Which is pretty darn fast, actually. And so you have to kind of think about then, what's the best way to make use of that time to be able to study at that pace. Obviously, you won't be able to rewatch every single lecture video for the stuff that came before midterm 2, because that would require you to listen to my voice for 24 hours straight for each of these 2 days. You'd be able to cover everything. So you'd have to think of some other way. So this is a good time to get tips from your TAs and from myself for how to review things efficiently. My tip for you, for example, would be to go through the lecture slides instead and flip through them very quickly, just to remind yourself of the terminology and the important concepts. And only if there's a concept that you're having trouble understanding, then turn to the videos and other resources in order to try to understand it better. Or just give me a call and we can talk about a topic that you're having trouble with as well. Right? That way you can get through this a lot faster than to rewatch all the videos at a slow pace. All right, so same thing for the new stuff. I'd say definitely spend as much time reviewing the lectures as you do doing problems, right? So for these review weeks, I'd say look at the lecture slides to just remind yourself of what we learned before. Maybe spend half an hour per week of lectures doing that, and then spend at least another half hour doing 2 or 3 practice questions from that week's practice exam questions, just to remind yourself of how to use that information. All right, so during that day when you're going through 4 weeks of lecture, then spend half an hour reviewing the lectures for a week, half an hour doing practice questions, then move on to the next week, half an hour reviewing lectures, half an hour of doing practice questions, just to solidify each thing after you review it, and do take breaks every half hour along the way as you switch things. Okay, that's kind of one of the ways you might be able to keep that pace of reviewing everything in 2 days in a way that's optimal in terms of maximizing your learning and practice without falling behind. All right, so this is the kind of pacing you need to figure out for each course, for each of these 4 or 5 days you set aside for studying, because if you go too slow and don't follow this pace, then you'll find yourself bleeding into the time you set aside for other courses as well. All right, so these are some newer tips for how to pace yourself for the final exam. And remember, pace yourself in such a rate that you can actually get enough sleep too. Other kind of tips to remind you of, do take the steps to eliminate your distractions now, and also this is a good time to ask for some help and support. So in terms of eliminating distractions, if you haven't already done so, definitely get someone to change your passwords on your social media, your Netflix, your video games, all that kind of stuff, just to get that out of the way, and so you don't lose any time with these distractions. But you can also try to create a bit more time and space and energy for studying by asking for some help from some family and friends who may not be doing finals coming up. So you can ask for people at work to maybe take over a couple of shifts, and you can help them back as soon as finals are over. Maybe ask your roommates or your parents to maybe drop off some food just so you can save some time from cooking. Maybe ask to not have to do your chores for a couple of weeks. Ask them to help you mow the lawn just so that you can get a couple more hours in for studying. All right, but most important tip of all though is there's gonna be like 2, 3, 4 weeks of preparing for finals, so you need to make sure you stay healthy and that you manage your sleep and stress levels very well. So for example, make sure that you have enough food, that you've packed some healthy snacks that you can snack on whenever you start getting a little hungry or tired, just so your brain is well fed and well nourished and you're not starving it several times a day. Make sure you're sleeping at least 7 hours a night. All right, now is not the time to build up sleep debt. So 7 hours a night. If you need more, then have 8 hours a night. Prioritize that so you can stay focused day by day. Otherwise, you're just going to waste time napping or not being focused anyways. And remember to take lots of breaks, right? Set a timer so you take a break, like take a mini break every 30 minutes at the most, where you just do some breathing exercises, walk around the house a bit, do like clean a couple dishes, and just take lots of breaks. And also for on the day-by-day way, make a to-do list so you can know what you're focusing on, just do one thing at a time and not think about everything else you need to do. And also at the end of the day, it's really easy to be stressed out and have trouble turning off your brain before going to bed. So make plans to just call a classmate or call a friend or family member just to talk out your stresses and clear your head and clear your stresses before you go to bed each night. All right, so there's a bunch of tips there, but do make use of them as you kind of dive into final exam time, and maybe use them again in future semesters as well. If you have any questions about this stuff, do feel free to give me a call. All right, so today's lecture is lecture 21, where we're just going to finish off looking at lac operons by digging a bit deeper and learning some better strategies. So in your last lecture video, you learned how to take a mirodiploid cell, so a cell that has two copies of a particular gene or operon, and even if it has different mutations in those two copies of a, say, lac operon, for example, we looked at how you might be able to predict what's going to happen in terms of its expression of structural genes in the presence or absence of lactose, right? So you learn to deal with quite a variety of different situations in terms of whether or not lactose was present, and in terms of the kinds of mutations that are present in one or both of these lac operons in the mirodiploid. So you did some good problem solving in this last lecture. So what we're going to do with this lecture is we're going to help you figure out what's the most efficient and reliable strategy for doing these problems. And you'll also take it one step further and look at how the lac operon responds to the presence and absence of glucose as well, which is a very important thing. After that, we'll also look at a couple of other operons, so an arabinose operon and a trypsin operon, in order just to help you understand other ways in which genes and operons might be regulated. All right, so quite a lot to cover in this lecture, so let's get started. So, last lecture, right, you did get some practice in figuring how different mutations affect gene expression in different combinations, actually. So when we're doing these simpler examples, you looked at how these different elements might interact, like, what happens when a super repressor meets a constitutive expression operator, that kind of thing. And along the way, you probably started to figure out your own strategies for solving these kinds of problems. It's almost like I've told you the basic rules for Sudoku, and you're looking for strategies for finding the answer most quickly, which is great, which is kind of a good kind of exercise in problem solving and flexible thinking and in finding strategies, because the more you're able to figure out your own strategies for a new problem, then the more efficient you're going to be than anybody else at solving that problem, which is super useful if you want to be employable and productive as a researcher in the future, to find strategies in the face of a new problem. For you guys, having done some of that and benefited from that, which is great. So if you're doing well in the clicker questions in the previous lecture, that's awesome. That means you're pretty darn good at problem solving. But now, just to make sure that everybody can get ready for the upcoming final exam, though, I want to help you develop the best strategy for approaching these problems. Okay, so you've had some freedom to figure out for yourself. Now I'm going to work with you to figure out the most efficient strategy for solving these problems, much like I did for the pedigrees, of showing you kind of the foolproof way to go. Okay, but we'll do it together, though. We'll figure out that strategy together instead of me just feeding it to you. All right, so let's go ahead and do that then. So what we're going to do is we're going to do two more example problems of these, predicting what the mirodiploid is going to do in a non-induced and induced situation, like when there's no lactose around and when there's lactose around. This time around, though, right, I'm going to get you to answer the question first, but as you're answering the question, so when you've paused the video and you're working out your guess at the answer, I want you to keep track the order in which you looked at each part of the operon to figure out what thing you should have looked at first and second and third and fourth. And then as you figure that out, also think about why. Like, why did you look at that part first and not another part? Okay, so after you've answered the original question, which is this one, we're going to look at it step by step and say, what should you have looked at first and second and third and so forth, and why? Okay, so keep that in mind. All right, so this is your question. Your possible answers are below, so you can pause the video and work out the answer, but keep track of the order in which you looked at the parts of the operons. All right, so pause the video and go ahead. All right, welcome back. So correct answer is C. So if you got that, that's great. If not, don't worry about it, because I'm going to teach you the method that will help you find the answer better next time around. Okay, so this is just a picture I'm going to add. But what I'm doing is I'm putting the correct answer at the bottom down here, and I'm also putting up the picture showing how different things can interact, how the I gene will make a repressor, and the repressor will bind to certain things, and that'll affect the expression, right? I'm just showing you a picture that we can look at when we're talking afterwards. All right, so step one, let's figure out what is the first part of the operon or gene that we should look at when we're trying to solve a problem like this, and why. Okay, so the question is, when you first started solving this problem, what was the first part of the operon you looked at, and why is that the best first thing to look at? Okay, so choose what answer you think makes sense. What is the first thing you should look at? Okay, so pause the video, think about it, and enter your answer, choose your answer. All right, welcome back. So I believe the best first step would be to look at P. So maybe pause the video again and think about why that might be the best first step. So pause now. All right, welcome back. So here's my explanation for why. Look at P first, because if there's a P minus for one of these operons, right, like we see in this second operon in this mirror diploid here, that means that you're not going to see expression of the structural genes for that operon, because if the promoter is really badly mutated such that RNA polymerase can't bind to it at all, then we're not expressing anything from this operon. We don't care what's happening with the repressor or the operator sequence or anything like that. If there's no good promoter, there's no expression, we can just cross off that entire operon right away. All right. If there is a P+, if there's a working promoter, then we should go to step 2 and see what else is happening with that operator, or with that operon. Okay. One thing to watch out for though. So here we see a P-, we're crossing off the rest of that operon, but do not cross off the I gene on that F plasmid in this case, because that's still important, right? That is not a part of this operon physically. It's not, its expression is not controlled by the same promoter sequence, and therefore this can still be expressed, and that protein can also affect the other operon as well. So when I say cross off this operon, I mean the P, the O, the Z, the Y, whatever genes are directly attached to that promoter, but not the I gene, because the I gene is separate. Okay, so that's step 1. Okay, look at it. If it's P-, you can cross off the rest of that operon, but leaving the I intact, because we still got to look at that. If there's a P+, we're going to have to go to step 2 and think of something else to look at next. All right, so step 2. Having looked at the P and we found that, oh, we have a P+ here, we're going to have to look at something else. What do you think we should look at next, and why? Okay, so pause the video now. All right, welcome back. So I think we should look at O next. So if you didn't choose O, now that I've told you it's O, I want you to pause the video and think of a reason why. All right, welcome back. So the reason why you look at O next is because if it's mutated in the way that makes it a constitutive expression operon, meaning that this operator is so messed up, so kind of mutated, that the repressor protein can no longer recognize it, cannot bind there to block expression from happening, then these structural genes, whatever is wild type, is going to be constitutively expressed. And it doesn't really matter what's happening with the I gene, right? If the operator is too messed up, the I gene could be a super repressor or a normal repressor. Either way, it's not binding to this particular operator sequence. So that's what you do when you see an O with a C, meaning that it's a constitutive expression mutation of the operator sequence. Then you can pretty much stop looking at the rest of this operon. As long as there's a P+ and OC, you know that all of the structural genes attached here are going to be expressed, whether or not there's lactose around, whether or not I is a super repressor, a plus or a minus, you're still going to see constitutive expression. Okay, so that's a good shortcut to finishing. But if there is a wild-type operator sequence, one that can still bind to a repressor protein, then you're going to have to go to step 3 and look further. All right? So notice that for this particular example, right, the first 2 steps already kind of gave us our final answer, right? The Z structural gene is never expressed because the only wild-type copy of the Z structural gene is in this operon, right? The one on the right, the one that's on the F plasmid here. But the promoter for that particular operon is badly mutated. It's got a null mutation, and therefore you cannot express the structural genes attached to this operon because RNA polymerase cannot bind, which is why for beta-galactosidase, the enzyme produced by that Z structural gene, the wild-type Z structural gene here, it's never expressed whether there's lactose around or not. Okay? Whereas, so that's the first step. Second step was looking at OC. So this tells us that the operator sequence is mutated and it cannot bind to the repressor protein, but there's a good promoter around. That means you're going to have no repression of this operon, and you're going to express its structural genes constitutively. And therefore, we see that permease, which is the only wild-type structural allele shown here, is going to, this Y plus allele is going to be expressed all the time to produce that permease, whether there is lactose around or no lactose around. Okay, so we already know exactly what's happening with the Z and the Y genes and their gene products, and therefore we're finished, right? So Y genes always is produced because we have a normal promoter and a constitutive expression operator. We don't have to go to steps 3 or so forth, 3, 4, 5, or whatever. Okay, so hopefully that's helpful. So this first example was a relatively short one, but it did help us to figure out the first 2 steps, right? It helped us look at P first and then O first and Y. Why we looked at those? Because if we see a P minus or an OC, we get the shortcut to a very solid and sure prediction and get closer to finishing our question more quickly. All right. If that was confusing at all to you, go back and rewatch this example again from the start first before moving on to the next one. But if that made good sense to you, let's keep going. All right. So next example, and this is the last one as far as helping us figure out the order of steps. Okay, so to help us uncover the remaining steps. So here is your question. Pause the video, and again, as you figure out your answer, keep track of what you're looking at and in what order. So start with P, then look at O, and then figure out and keep track of what your remaining steps are. All right, so pause the video and do that now. All right, welcome back. The answer is B, so everything is minus. So now I'm going to move the correct answer down here again, and I'm going to show this figure here. So one thing you might want to do is, for the previous example, go back and look closely at the picture once you've finished watching that part of the lecture, because it does visually show the exact mirror diploid that we're looking at for example 1. And this picture here shows the exact genotype for this mirror diploid, and shows you schematically, where the repressor protein can and cannot bind, whether there is any enzyme being produced and why, whether RNA polymerase can bind or not, whether the I gene is making repressor or not, right? So this figure is actually quite instructive and quite a good way to interpret your answer afterwards. But in my explanation, I'll focus on this because this is what you'll see in the exam. But for those of you who are visual learners, this figure here is a perfect match for these examples that I'm giving you right now. So do make use of it if you're a visual learner, if you like. All right, so step 1, like I said, look at P. And once again, that's very helpful because we see that there is a P minus here, right? And that tells us we can cross off the rest of the operon, keeping the I, just in case that might somehow show a trans effect on the other operon. Okay, so you can cross off those structural alleles. Even though they're both wild type, we're not going to be able to express those because the promoter is too messed up. But we do see a wild type promoter in the second operon, and therefore we're going to have to go to step 2 for that one. Step 2 told us to look at the operator sequence, right? And as much as we're done with this one, I'm just going to, for argument's sake, let's pretend that we forgot that we're supposed to ignore the rest of this operon, and we went and looked at the operator sequence. Notice that, okay, this one has a constitutive expression operator, meaning it's mutated and the repressor protein can't recognize it. But we can actually still ignore that because notice that if the promoter doesn't work, right, it doesn't matter that there's no binding of the repressor protein here. It doesn't matter that these structural genes are wild type. If the promoter is not working, we're not going to get transcription no matter what, which is why this was kind of a waste of time. We didn't have to look at the operator sequence. But I just wanted to pause here and point out
that in step 2, we're not going to look at this operator that we crossed off, because whatever this operator is, it doesn't matter as long as P is minus. Okay, so that's why step 1 is to interpret P first to avoid having to look at any stuff that doesn't matter anymore in this operon. Okay, so having done that, let's focus on the operon we're supposed to focus on. Is okay, so the one that has P plus, the one that got past the operon that got past step 1. Let's look at the operator sequence. The operator sequence has a wild type operator sequence. So as we said previously, we can't jump to the ending now because it's not an OC sequence. So we're gonna have to look at step 3 and figure out what the heck step 3 is gonna be. Right, so if looking at P and O doesn't help us finish the question, we're gonna have to look at one more thing at least. So what should you look at next? So pause the video, make a decision. All right, welcome back. So I is what I suggest. So if you didn't choose I on your own, I want you to pause the video and think about why I'm saying that I should be next before coming back. So pause the video. All right, welcome back. Okay, let's look at it bit by bit. So let's say that there is at least one super repressor allele for this gene. All right, then remember that in the allelic series, the super repressor is dominant, the most dominant one. Then the next dominant one is the wild type repressor protein, and then the most recessive is I minus, the null one that doesn't make any active repressor. So if you have a super repressor, right, the super repressor protein that it makes will go around and permanently bind to all the operator sequences that are still wild type, and block them from being expressed permanently, right? So remember that this I gene acts in trans, which is why you're going to have to look at both of the copies we have and see which ones are present, right? As long as one of these two is a super repressor, then any wild type operator sequence will be bound by that super repressor and stop expression altogether. Okay, if it's a constitutive expression one, then no repressor will bind to it, no matter what type. Okay, so this is one shortcut to the ending: is if you see a super repressor allele, find any wild type operator sequence and know that that's going to be blocked permanently from now on by the super repressor protein produced by that one super repressor allele, or both. Okay, another way to shortcut to the end is if you only have I minus allele. So if you're homozygous for the most recessive allele of my I gene cannot make active make repressor protein, and my other one also can't make repressor protein, that would mean that there's no repressor protein around at all. If that's the case, then then you're not going to be blocking this operator sequence at all. So as long as an operon has a wild type promoter and there's no repressor protein to block it at all, then you should be expressing whatever that promoter is attached to. Okay, so since there's no repressor, if there was no repressor protein being produced, then your operons will be constitutively expressed, assuming that your promoter sequence is still wild type. Okay, so those are the two kind of possibilities that will get you to an ending more quickly. But if instead you have at least one wild type I gene and no super repressor, right, then you're expecting normal expression of your operons that still have a normal operator sequence. Okay, so in this case where you have a wild type repressor being produced and no super repressor, then whether or not an operon is turned on or off will depend on where the lactose is there, right? So remember that this repressor protein will bind to the operator sequence when there's no lactose around, but it'll be taken off when lactose is present. Okay, so a tip to remember is that you should always look at both I genes. Even if you crossed off one of these operons, look at both I genes, because remember that I genes make a protein that can act in a trans way. It can go to either operon and act on it. Okay, and also remind yourself that this is the allelic series. The super repressor is dominant over the wild type repressor, which is dominant over the null mutant repressor. So in this case, for step 3, notice that we do have a null mutant repressor gene here, and the repressor allele here. So that's not going to make any repressor protein, or not one that does anything anyways. But we do have a wild type repressor gene here, right? And that is the more dominant one than the null mutant. So what matters right now is that we are making a normal repressor protein and one that can respond to the presence and absence of lactose. So we're going to have to move on to step 4. Okay, so these super repressors and homozygous for the null mutant will get you to be able to finish the question more quickly. If you have a wild type repressor gene and no super repressor, then you're going to have to go to step 4. Okay, so what should we look at for step 4? Yeah, so pause. This is not a clicker question, so pause the video and think about what to look at next. All right, welcome back. So at this point, we should take a look at the structural genes, right? Because we want to see which enzymes are being made by the operon that we're still looking at by step 4. So we're looking for any structural genes that are actually still wild type and therefore can make some active enzyme. So in this example, right, this second operon, which has the working promoter, has the wild type operator sequence, it doesn't actually have any wild type structural genes. Notice that both of the structural genes are mutant. This might have been a mistake by whoever set up this mirror diploid in that they didn't distribute one wild type allele for one gene and a different wild type allele for a different structural gene among these two copies of the operon. But in either case, the only operon that has any chance of being expressed does not have any wild type versions of the structural genes we're looking at. All right. So as a result, the Z gene, the only wild type copy we saw here, is not going to be turned on because of the mutant promoter. So it's minus minus, and we're hoping to see a permease being expressed on this side, but its only wild type copy is again in the broken promoter operon, and this side that could have been expressed when lactose is around isn't going to be expressed because it's a mutant copy of the gene. Okay, so if you want to help yourself remember these 4 steps in the order to look at them, the abbreviation is POIG, which doesn't make much sense. So POIG, right? But the letters make sense. You're looking at P first, then O, then I, and then G I used as just an abbreviation for structural genes. Okay. So to help you remember that a bit better, if you're a fan of The Walking Dead and you watched the first season, you can avoid trouble by putting on infected guts. Or a poig, okay? Hopefully that's not some sort of weird insult in some language, okay? But putting on infected guts should be probably safer than poig. All right. If you have any questions, do ask your TAs or myself anytime. But let's move on because I'm going to add a couple more letters after POIG actually. So let's look at how glucose affects lac operon expression, okay? So to help you understand why glucose matters, right, I want you to answer this question. So what effect should the absence of glucose have on lac operons when lactose is present? So in the situation where there's some lactose around and I'm going to take away the glucose, how should that affect lac operon expression? So I want you to choose your answer and also to write down your best justification for that answer. Okay, so go ahead. All right, welcome back. So the answer is A. So lac expression should be promoted with an activator when there's not a lot of glucose around. So here's just a picture of what an activator is that will promote lac operon expression. I'll explain how that works in a little bit. So why does this make sense, right? Why is increasing expression of the lac operon make sense in this specific situation outlined up top? All right, so maybe pause the video and think of a good answer to this question. Pretend this is an exam question you came across. How might you answer it? All right, so pause the video now. Right, welcome back. So let's take a look at this bit by bit, right? In the absence of glucose, if there's no glucose around, glucose being kind of the simplest sugar that you can directly use for glycolysis and or cellular respiration. When there's not enough glucose around, as the main and most energetically efficient energy and carbon source, it makes sense that you are then going to need to use other sugar sources, ones that might not be as energetically efficient, but you need the energy. And so in this situation where there's not enough glucose, you're going to need to more efficiently use lactose, using these, with the help of these lactose enzymes, as an alternative source of energy. Okay. On the other hand, if glucose is already abundant, right, then you would expect to see much less expression of the lac operon. Here's that's it. So maybe treat that as a practice exam question as well, and think about why it is that if there's lots of glucose around, that you would see less expression of the lac operon. So yeah, pause the video and try to figure that one out. All right, welcome back. I'll give you a bit of a hint. So there's a couple of reasons you can look at, both the proximate and the ultimate reason. So if you've taken BISC 102 with me, you'll know what I mean by that. If not, don't worry about it. That's just a hint for some people who have taken 102 already. All right, so pause the video and try one more time to try to answer this question in two different ways for why you would expect less expression of the lac operon when there's lots of glucose around. All right, welcome back. So, ultimate explanation. So why is it kind of better for the fitness of the individual if you decrease lac operon expression when there's lots of glucose around? Well, the ultimate explanation is that I kind of hinted at pretty heavily in my explanation of the earlier questions, but digesting lactose actually takes more time and energy than glucose, right? So using lactose as an energy source kind of costs you some energy efficiency. Like you don't get quite as much net amount of energy gained as if you digested glucose directly. So glucose being the better energy source in terms of net energy gain. If there's lots of glucose around, you should prioritize the use of glucose and not waste as much time and energy for digesting lactose. Okay. The approximate reason, so the mechanistic reason why you might not get as much lac operon expression when there's lots of glucose around is that when there's lots of glucose around, this activator protein that would crank up the expression of the lac operator, that's not binding to the lac operator. It's not, the activator protein is not present. Okay, so we'll look into the details of that. That's just kind of to help you set the stage for why glucose matters and how you expect the glucose presence or absence to affect lac operon expression. So hopefully by now you understand that when there's no glucose around, you'll want to express this more and get more energy from lactose. But when there's lots of glucose around, you will express this less because you don't need lactose as an energy source as much, and you would focus more of your energetic efforts on using glucose instead. Okay, so let's take a closer look at how this activator works and how positive regulation works. Okay, so this is a good chance for you to kind of compare positive and negative regulation with which is carried out by activator and repressor proteins within a single operon. Okay, so negative regulation, we'll focus on that side of things first because that's the side you already know well. Okay, so in negative regulation, we see a repressor protein, the one that's made by the I gene, the wild type I allele. The repressor protein normally binds to the operator sequence, right? And it binds between the promoter region and the actual coding sequences. And when it's bound there, it blocks RNA polymerase from binding and therefore blocks transcription, right? So we call this negative regulation because the binding of this regulatory protein represses DNA transcription. It represses gene expression. Okay, negative because binding equals less expression. So positive regulation, on the other hand, must be that binding increases expression of the gene. So let's say that the repressor protein is not blocking transcription, right? So there is lactose around. This is not affecting transcription at all. You would still see very little transcription happening unless the activator is binding to the binding site. All right, so basically, if you want to see full expression of this operon at the maximum normal levels, then you need the activator protein to be bound to the DNA at the specific site, at the activator binding site. That activator binding site happens to be beside the promoter, which makes sense because if it's going to be helping the RNA polymerase to bind to the promoter more efficiently in order to increase transcription, then it should be close to the promoter sequence. Okay, so notice we have two different proteins. The one that's involved in negative regulation will bind in such a way that it blocks transcription completely. But if it's not being blocked, how much transcription occurs depends on whether the activator is there. If the activator is there, you get lots of transcription. If it's not there, you'll get very little transcription. Now, depending on your figure looking at, this one shows no transcription. That's not exactly accurate. You still do get some amount of RNA polymerase binding and some expression of the structural genes. So for the purpose of this course, all right, we'll assume that the repressor protein will block transcription entirely when it's there. when it's bound to the operator sequence. Whereas for the activator side of things, when the activator is there, you'll get full normal expression of the operon. If the activator is not there, you will get a small amount of transcription. Okay, in both senses, it's a little bit of a gray area, but this is a more kind of complete blockage, relatively speaking, and this is you still get some amount of transcription in this situation here, but way more if you add the activator. Okay, so overall, as far as comparing positive versus negative regulation, right? In negative regulation, the binding of the regulatory protein decreases transcription. That's why it's negative regulation. Whereas in positive regulation, the binding of the regulatory protein increases transcription. Okay, so that helps you understand that both activator proteins and repressor proteins, they're both specific examples of regulatory proteins, proteins that bind to DNA and therefore affect its expression, affect the expression of the associated gene or operon. All right, so both of these regulatory proteins are called regulatory proteins because they regulate the expression of genes or operons, and it does so by binding to DNA sequences at their specific sites. So the repressor binds to the operator sequence, the activator binds to the activator binding site, a pretty not creative, but at least easy to remember name. Okay. Now, besides that, the reason why they're useful is that besides the fact that they can turn off or crank up expression of the gene, is that they're doing so in response to environmental conditions. They're helping us to turn off the operon when there's no lactose to digest. They're telling us to get more expression of operon when we need it more. Right? So they have to sense environmental conditions. And so the way they do that is by binding to specific effector molecules using the allosteric site. So they have a DNA binding site that will either decrease or increase expression of the operon. But there's also an allosteric site that's binding to something from the external environment or a intermediate signal that originated from the external environment in order to respond to the environment in the right way. So when these effector molecules bind to the allosteric site of these regulatory proteins, it changes the shape of the protein such that its affinity, its ability to bind to the DNA gets changed. All right, so let's look at this in a little more detail. Let's start with the one we know well, okay? So focusing on the lac repressor protein, the one that's made by the I gene, right? What is the effector molecule that binds to this lac repressor? Okay, here are your choices. So pause the video and make your choice. All right, welcome back. If you weren't very sure, I'll give you one more hint before I reveal the answer. So here's a hint. This effector molecule that binds to the lac repressor is lactose. Right? Remember, the only other thing that binds to the repressor protein besides the operator sequence of the DNA is lactose. Okay, so knowing that this effector molecule is lactose, try answering this question one more time or confirm your answer. All right, welcome back. So the answer is D. All right, so notice that when lactose binds to the repressor protein, then the repressor protein is no longer as attracted to the operator sequence because it's changed its shape, right? And therefore it comes off of the operator sequence and stops blocking lac operon expression. Okay, so adding lactose removes the repressor, therefore you get more transcription than before, which makes sense because when lactose is around, it makes sense that you would want to get some enzymes to digest it. All right, so now that you kind of used the repressor protein that we're quite familiar with to figure out what the effector will be, let's take a look at the lac activator protein and what its effector molecule might be instead. So here's this similar question, but focusing on the lac activator. So it's a somewhat more difficult question, so take your time and make your best guess and think about how you might write your justification for this question, for your answer if this was an exam question. So pause the video and choose your answer and try justifying it. All right, welcome back. So the answer is B, all right? That in this situation, and as is kind of shown in this picture as well, when the effector molecule binds to the activator protein, it actually makes it more attracted to the to its binding site on the DNA, and causing it to bind on and increase the amount of transcription happening, right? So notice that overall, like based on this picture here, in both cases, when the effector is added to the cell, it ends up increasing lac operon expression, right? It does so either by lactose binding to the repressor protein and removing it and getting it out of the way, and therefore allowing transcription to happen, or by binding to the activator and causing it to increase its binding to the activator binding site, and from there allowing it to promote the binding of the RNA polymerase and getting it to do more transcription. All right. So the effector molecules in the lac operon, both of them end up increasing transcription of the operon structural genes, but just in different ways, either by removing the repressor or adding the activator to the DNA. All right. Now, what the heck is the effector of the lac activator? So what is this effector molecule? It was lactose here, but it's something else here. So I want you to think about what logically makes sense in this situation. So pause the video and make your choice. All right, welcome back. Before I give you the answer, I want to give you a hint and another chance to change your answer if you want to. So think about how glucose should affect lac operon expression in general. Okay? If you added glucose, what should happen to lac operon expression? If you took away glucose, what should happen to lac operon expression? Keep that in mind and double check your answer. So pause the video now. All right, welcome back. So the answer is D. Okay? And it makes sense that you want something that when there's not enough glucose, it builds up, and that's going to signal this activator to bind on and start expressing this more. Okay, so that makes sense because you don't want it to be glucose itself, right? If it's glucose, that would mean that, oh, there's lots of glucose around, there's lots of the easiest possible sugar to use. You don't want glucose to come along and turn on the activator for expressing the lac operon. That wouldn't really make sense. If you have glucose around, you don't want to waste time making enzymes that you don't need. What you actually want is the opposite of that. You want it so that when there's lots of glucose around, you leave this activator off and don't do much transcription of the lac operon because you don't need it when there's lots of glucose around. You want instead for this activator to stay off until there's not enough glucose around. You want it so that when glucose is not around, whatever accumulates in that situation, that is what should finally turn on the lac operon. This is in this situation that we actually want to activate this operon in order, or express this operon in order to start using lactose more heavily as an energy source when glucose is not around. And it's a little convoluted to think about something building up when glucose is not around, but you'll see what that is in a moment. Okay, but first to finish off here, so let's focus on what the lac activator and its effector is. All right. So the lac activator protein itself, the regulatory protein that actually directly binds to the DNA, is called CAP, which stands for catabolite activator protein, which again seems like a lot of syllables, but it kind of makes sense when you think about it. It is an activator protein, so it's a regulatory protein that when it binds helps to activate genes or increase the expression of genes or operons. And catabolite refers to catabolic reactions, which are reactions where you are breaking apart something in order to release energy, which makes sense. So we want to break apart lactose to release energy, and this is the activator protein for helping that to happen more quickly and immediately. All right, so that's our activator protein, that's a regulatory protein. And then the effector that binds to it is cyclic AMP. Again, a lot of syllables, but AMP is adenosine monophosphate, right? Which is basically like a broken down, used up version of ATP, which would be adenosine triphosphate. All right, so let's talk about why this is that effector molecule we're looking for, the thing that builds up when there's not enough glucose around. So normally when there is enough glucose around, then glucose gets broken apart by glycolysis and the rest of aerobic cellular respiration, and therefore you're able to recharge ADP into ATP. So ATP is adenosine triphosphate, so it's an adenosine base but with 3 phosphate groups instead of just 1, and that is a very kind of unstable, highly energetic molecule that we use to provide energy for many chemical reactions in our cells, and it's kind of the energy currency of our bodies. And usually when an ATP releases a single phosphate group and becomes ADP, so from triphosphate to diphosphate, from 3 down to 2, then we've released the energy, then the ADP basically using things like ATP synthase and a substrate level phosphorylation, so two different things that happen during aerobic cellular respiration, you would be able to put that third phosphate back on to the ADP to turn it into ATP again. So usually when there's enough glucose around, you make ATP as you break apart the glucose and release the energy from that glucose. And when you break apart ATP, it becomes ADP and a single phosphate group. And then you use the energy released from glucose to put that phosphate back on, to turn it back into ATP. So it goes back and forth between ATP and ADP. But if there's not enough glucose around to provide energy to recharge the ATP again and again, then some of the ATP gets broken down further and gets converted into AMP, so adenosine monophosphate. So instead of just losing one phosphate group and becoming ADP as it normally does, it loses another phosphate group and becomes AMP, okay, adenosine monophosphate. So this only starts appearing when there's not enough glucose to recharge the ADP back into ATP very quickly. All right, so this is our sign that, we're not getting enough energy through glucose. We're going to have to start using something else, otherwise we're not going to have enough ATP to stay alive. So cyclic AMP is our effector molecule. This is what's going to bind onto our catabolite activator protein, our CAP, right? And these two join together, which we're going to call the cyclic AMP CAP complex, which is basically just the names of these two things with a dash in between or a hyphen in between. That is the activator complex that's going to bind near the promoter of the lac operon and therefore increase transcription a lot. Basically helps the RNA polymerase to bind to the promoter way more effectively and thereby make way more transcription and translation happen of the operon's structural genes. Okay, so that is our regulatory protein, is CAP, and the effector that turns it on and gets it to bind onto the promoter and start increasing transcription of the operon is the cyclic AMP CAP kind of complex. Cyclic AMP is the effector molecule, just like lactose was the effector molecule of the lac repressor. Okay, so basically, so cyclic AMP increases expression of the lac operon, and that's great because if we're low on glucose and we're running out of cellular energy, then we want, and there's enough lactose around to take off that repressor protein, meaning that there's some lactose around to digest, then by using the cyclic AMP to turn on CAP and to activate this operon, we're going to transcribe and translate a lot more of these enzymes, which we're going to need to use up that lactose quickly and get some more energy back into the system, into the cell. Okay, so that's what happens when there is not enough glucose around and lactose is present, and we're going to want to get the enzymes necessary to use that lactose as soon as possible. Now, if there is lots of glucose around already, right, then you're not going to be making cyclic AMP, right? Because you have lots of glucose around just to recharge the ADP back to ATP really quickly. So if there's no cyclic AMP being made, then there's nothing binding to the CAP regulatory protein. So without cyclic AMP bound to it, CAP by itself does not bind to the promoter region at all. So that means the activator can't bind to the DNA, and therefore RNA polymerase has a lot more difficulty binding to the promoter region, right? So very rarely is it able to bind to the promoter well enough to express or transcribe the structural genes once, and therefore overall you get very little lac operon expression. Okay, so almost none, but not quite none. So just very, very low expression. All right. So remember, for this course, we're going to count. So the situation where there's no repressor, and no activator bound, we're going to assume that there's still a small amount of expression, very small amount of expression. Whereas if the repressor protein's bound, then that blocks transcription altogether. Okay, that's kind of the slightly simplified version of the gray area we're going to stick with for this course. Okay, so click a question. So when glucose is abundant, so when there's lots of glucose around, but there's no lactose around, how much lac operon expression would you expect to see for a cell that has this genotype? Notice that this isn't even a mirror diploid. This is just a cell that has one lac operon. Okay, so once you've decided, I also want you to think about your justification. So again, imagine that this is an exam question, and after giving your answer, that you're supposed to write a clear justification for it. So pause the video and get both your answer and justification done first. So welcome back. So the answer is A, no expression. So if we're going to look at this one piece at a time, so the promoter is working normally. We have a wild type I allele, which means that the normal repressor protein is going to be around, looking for lactose to be present. And also we have a normal operator sequence that the repressor is going to be bound to normally if there's no lactose around. Okay? So in this case, we saying that lactose is absent. So there's no lactose inducers around to bind to that normal repressor protein to get it to come off of the the operator sequence. And so this repressor protein is bound to the operator sequence, blocking transcription from happening. There is no lactose around to take it off. Therefore, with the repressor blocking lactose expression, we're basically not going to get any transcription or translation of the structural genes. All right, that's why we see no expression. So logically, this makes sense because why would you bother making any of these enzymes if there's no lactose around to digest anyways, right? So logically, it makes sense. And also, we mentioned just before this that when there is a repressor bound to this wild type operator sequence, that's going to permanently and not permanently, but it's going to completely block transcription from happening. So no expression. Now let's ask a slightly different question. So let's say that we still have lots of glucose around, but lactose is now present. We now have lactose around in the cell and outside the cell. So how much lac operon expression would you expect from this same cell with the same genotype? All right, so pause the video and come up with both your answer and your justification as if this was an exam question, and then restart the video after that. Go ahead. All right, so the answer is B, very little expression. So now that we have lactose around, the lactose acting as the effector molecule or the inducer, it's going to bind to the repressor protein. And when the repressor protein is bound to this inducer, bound to lactose, its effector molecule, then its DNA binding site changes shape and it is no longer as attracted to the operator sequence and it comes off. Right? So therefore, RNA polymerase can finally bind to it and start doing gene expression, start transcribing these structural genes. However, so basically the repressor is not blocking transcription anymore. We can actually get some gene expression. But because there's a high concentration of glucose around, and the higher the concentration of glucose, the lower amount of cyclic AMP that's around. Without cyclic AMP, the CAP kind of activator protein cannot bind to the DNA, and therefore RNA polymerase is not as efficient at being able to bind and start transcription. So there's very little lac expression when cyclic AMP doesn't have, when the cap protein doesn't have cyclic AMP to activate it to make it more able to bind to the DNA. All right, so final question, changing one more thing. So we have lactose present again, but now we're going to take away the glucose. And I want you to think about how that will affect expression of the lac operon. Now notice this time I'm changing the genotype, right? I'm using green to highlight new things. I'm giving you a proper mirror diploid this time with 2 copies of the operon and a bunch of different mutations. So I want you to analyze this carefully and tell me how much expression would you expect to see of the lac operon in this situation. All right, so go ahead. So this is a harder question because you actually have to fully process this mirror diploid, but hopefully you remembered your putting on infected guts order in which we should be looking at each part of these operons. So the correct answer is C, normal amount of expression. So with that answer in mind, let's go through this step by step. Remember, the first step is to look at the P, the promoter regions of each operon. So we do find that the second copy of the operon, the one that's on the plasmid, does have P minus, meaning the promoter region is very badly mutated, and so we can ignore the rest of that operon, but we're going to maintain we're not going to cross off the I gene because we may need that. Let's take a look at that later on. Second step is to look at the O, the operator sequence or region of each of these operons, but ignoring this one because we're already finished with the operon on the right, but the one on the left does have a normal operator sequence. And so we should move on to step 3, which is to look at the 2 copies of the I gene that we have. Now, even though this I gene is a null mutant that doesn't produce a repressor that can bind to the operator sequence, over here we do have a normal wild type I allele, and therefore we do expect that a normal repressor protein can be produced, and therefore we're expecting normal regulation of this first operon because we have a normal operator sequence and we have the normal regulatory protein produced, even though the regulatory protein is being produced by the second copy of the operon that's found on the plasmid. Okay? So next, for the fourth step, we're going to look at the structural genes, and we find that, oh, look, we have not just Z and Y, but also A. So all three of the structural genes are represented in this picture, and all of them are wild type for this operon, which is great because we want to see whether we're going to be able to digest, absorb and digest lactose here. So that's the four steps, but in this case, we're actually specifying exactly whether or not glucose and lactose are present. So let's look at those one at a time, starting with lactose. Because we'll look at lactose first, because if there's no lactose around, then the repressor is going to bind here and we're not going to get any expression at all. So we're going to look at lactose first. So lactose is present, which is great because that means that the lactose is going to bind to the repressor protein, get the repressor protein off of the operator sequence, so there's no longer blocking transcription. So there's the possibility that we're going to actually express this gene. It's just a question of how much. So then we're going to look at glucose after that, then knowing that there's enough lactose around to get rid of that repressor. So since glucose is absent, then ATP is going to get broken down past the ADP stage all the way to become cyclic AMP. So that cyclic AMP is going to bind to the cap protein, which will help it now to bind much more strongly to the promoter region of the lac operon. And from there, that cyclic AMP cap complex can help RNA polymerase to bind the promoter more efficiently and therefore get way more transcription than if the activator was not there. All right, so notice that our full set of steps now starts with POIG, right? And then at the end, we're going to look at lactose first, then glucose, right? So the full set of steps that you should follow in order to solve these problems with the lac operon as efficiently as possible is put on infected guts. Looks gross, if that helps you remember it. I really loved Walking Dead, and I was watching lots of it when I wrote these lectures. So if you're not sure what Walking Dead is, it's just a zombie show. It's pretty good. Check it out when you're taking a break later. All right, but that's pretty much it for the lac operon. That should give you everything you need. Now, the more of these practice questions you do, the better you'll get at this. So remember that with exams, speed can matter. The more you practice using certain tools, the more quick, efficient, and accurate you get with them. And so you'll want to do more practice. If you run out of practice questions, I want to give you a recipe for writing your own. All right, so you can make up questions, share them with your study group, and always remember to practice writing explanations too. That's where strong students and average students both lose a lot of marks, is not knowing how to explain your answers, even if you can find the correct one. There's a lot of marks lost with bad explanations. All right, but here's the recipe. So start with this basic genotype, right? Whether or not you include the A gene is up to you. Usually you just need two. One active, one that's wild type for one operon, and one that's wild type for the other one, but you can try all three if you like. And then from there, change a bunch of the superscripts, okay? So you start with all of them being plus, but choose as many of them to change as you like. In terms of how you can change them, starting from left to right. For the I gene, remember you can either have it be wild type, null mutant, or super repressor. For the operator sequence, it's either wild type or C for constitutive expressions, either one or the other. And then all the other ones can be either plus or minus. Okay. After that, you can also, if you want, specify whether lactose is present or absent, and also specify whether there's lots of glucose around or no glucose around. All right. So that sets up the conditions, but then you should still be asking specific questions because, in my exams, I tend to ask you very specific questions. So you're focusing on one thing at a time and not the whole big picture and giving me everything. So ask a very specific question about the amount of specific enzymes being produced, or whether certain regulatory proteins are active or not active. You can ask what the effectors are and what they're doing. Are they present? And what happens if you take it away? And you can also ask yourself or each other what kinds of justifications or explanations you can give for each of your predictions as well, just because that's something that a lot of students struggle with and need practice with, which is justifying their predictions. So not just knowing how to do the work, but having good reasons for it as well, based on the concepts and what's actually happening with the operon. All right, so that's your recipe for making up your own questions. So make as many as you need and share them with each other in your tutorial groups and your study groups in order to get more practice with these kinds of problems. All right, So next, let's look at another operon, a different operon, not the lac operon, but this is also going to be another one that has to do with catabolizing sugar in terms of breaking down a different type of sugar as an energy and carbon source. So we're going to look at the arabinose operon. So arabinose being another sugar like lactose, but different. Okay, so this operon has a structure that's quite similar to the lac operon in many ways because it's performing a pretty similar function for E. coli cells. In that when there's no glucose around, we're going to want to express this operon in order to digest and use a different sugar, arabinose, as an energy source. The only difference is it's looking at a different sugar, and there's a few structural and functional differences as well. Right? For starters, one difference is that all of these structural genes are going to make enzymes and proteins that are needed for absorbing and digesting arabinose. Okay, so let's compare these two. You'll notice a lot of colors coming up. I'm basically been sticking to pink for the things describing what's happening for the new arabinose operon that we're looking at, and blue for the lactose operon. If you're confused by the colors, don't worry about it. Just make it black and white on your own screen if you like, and go from there. But I do find it kind of helpful for myself. All right, so more colors. I'm going to use green to help highlight the things that are the same between these two operons, and red to highlight the things that are different between our pink arabinose operon and our blue lactose operon. I've had complaints about there being too many colors, but personally, I think it helps me to visually separate the similarities and differences and so forth. Okay, similarities. So both of these operons have 3 structural genes each. Different letters, symbols used for each operon's structural genes. So arabinose operon has structural genes B, A, and D, whereas the lac operon has Z, Y, and A. Okay, so fairly simple differences. Obviously, these structural genes are going to be different for these two operons, and they have different symbols to reflect that. Another similarity is that the point of these enzymes, the purpose of them is to catabolize sugars, to allow the cell to absorb and or digest these sugars to get energy from them. But a major difference is that the sugars that they're catabolizing, that these enzymes are catabolizing is different. So arabinose for this operon, lactose for this operon. In terms of the control sites, the sites that control whether or not the operon is being expressed and how much, they're located just upstream of the structural genes right beside them for both of these. But there's some differences in terms of where they're located. So notice that for the arabinose operon, the operator sequence is found upstream of the promoter, whereas for our lac operon, remember that the operator sequence is downstream of the promoter, kind of getting in the way of RNA polymerase. Okay. Another difference is that there's this extra region called the initiator region that's found between the operator and the promoter sequence, and you'll see what that's for later on. But notice that, so those are some of the major differences. So there's still an operator sequence in both. It's just that this one's downstream, this one's upstream, and there's this extra initiator region here. Also for both of them, so green for similarities, there is another gene outside of the operon that's not part of the operon itself that codes for the regulatory protein. So the I gene is the gene that's outside of the lac operon itself, but it's important, and we consider it when we look at the lac operon because it produces a regulatory protein, the lac repressor, which binds to the operator sequence. And then in the arabinose operon, outside of the arabinose operon, there is another separate gene, the C gene, that codes for its regulatory protein, the RSC protein. Okay, so notice that the gene has a different symbol, C instead of I, and the protein that it makes is different, RSC protein instead of the lac repressor protein. Okay, so those are all the green similarities and red differences between these two operons. All right, so with those in mind, let's look at how the arabinose operon is regulated and compare it again to the lac operon, just so you have this foundation to draw upon. Okay, so in terms of negative regulation, in terms of blocking transcription when there's no lactose or no arabinose around, lactose and arabinose operons are pretty similar. So starting with the lac operon first, just as our foundation, when there's no lactose around, then the lac repressor protein is going to bind to the operator sequence, and that blocks transcription from happening. It blocks RNA polymerase from binding and transcribing the structural genes. So notice that the arabinose operon is pretty similar. When there's no arabinose present, right, so when arabinose sugars are completely absent, then the RSC protein will bind to the operator sequence, just like the repressor binds to operator sequence, and that results in the blocking of transcription. But just notice that the difference is that there's a slightly different blocking mechanism, right? Whereas the repressor protein binds to operator sequence, kind of where the RNA polymerase is trying to bind the promoter, it just kind of gets in the way of RNA polymerase binding directly by being there, right? The RC protein does things a little bit differently. The RC protein is already bound to the initiator sequence all the time, but when there's no arabinose around, it also binds to the operator sequence. And so the way that it blocks transcription is by forcing the DNA to form this loop, right? By forcing it to form this loop, the DNA is actually blocking the promoter region and not allowing the RNA polymerase to bind. So bending the DNA into a loop will block RNA polymerase from binding. It also blocks this CAP-C cyclic AMP complex as well, which we'll talk about in a little bit. All right. So overall, similar idea. When the sugar that they're supposed to digest is absent, the protein binds to the operator sequence, and whether it's directly or indirectly by bending the DNA, we're going to block RNA polymerase from binding to the promoter. Okay, so that's negative regulation. Looks fairly similar between the two, just slightly different mechanism. So now that we're done looking at how negative regulation of the arabinose operon, let's look at positive regulation. Let's look at how we can activate more expression of the arabinose operon and compare it to how that works in the lac operon. So again, let's start with the lac operon just as a review and as a foundation before looking at the arabinose one. So for the lac operon, when lactose is present, and therefore the repressor is out of the way and we don't have to worry about it, and glucose is absent, so we really want to express the lac operon a lot so we can digest that lactose for an energy source. What happens is, well, in terms of the lactose being present, that's going to bind to the repressor, get it off of the operator sequence, and therefore stop blocking transcription from happening. But when glucose is absent, that's going to make cyclic AMP start to build up as ATP gets used up further and further. And so that cyclic AMP binds to CAP, and together that complex binds near the promoter of the lac operon and basically helps RNA polymerase to bind and really increases expression of the structural genes from the lac operon. Okay, so basically the way we crank up the amount of transcription happening is by letting cyclic AMP build up, bind to CAP, which together bind to the promoter region, and that helps transcription happen way more quickly. So let's look at arabinose then. So when arabinose is present and glucose is absent, right? Same situation but for arabinose. So arabinose binds to the RC protein, right? And again, that causes it to detach from the ARA operator sequence. And notice that by detaching from the operator sequence, the DNA is no longer looped around, is no longer blocking the promoter region. Okay, so basically no longer repressing, just like this is no longer repressing. All right. Now, in terms of the cyclic AMP, so let's say that still there's no glucose around, cyclic AMP is building up. That's going to bind to CAP again. And the cyclic AMP CAP complex will also bind near the promoter of the arabinose operon, and again, greatly increases the expression of the structural genes. So you get a lot more mRNA from these structural genes, and you'll be getting way more enzymes for digesting arabinose as well. So notice that both operons use the exact same regulatory protein, CAP, for positive regulation, and that responds to the exact same effector molecule, the same activator molecule, which is cyclic AMP for positive regulation. Right? That seems like a coincidence, but it's really not, because this response, this activation of these operons is in response to the absence of glucose. And that makes sense that when there's not enough glucose around, then the cell's basically saying, we're going to have to use something else. So it makes sense to have the same activator complex turned on and ready to go. And it's going to go ahead and activate a very robust expression of the sugar catabolizing operon. So if there's lactose around, we're going to make lots of these enzymes to use lactose really quickly. But we're also going to be careful, right? If there is none of that target sugar around, if, for example, there's no arabinose around, then maybe you'll get lots and lots of expression of the lac operon, but no expression of this one. This is going to be blocked by that looping of the DNA, because if there's no lactose around, you don't want to be wasting time and energy and resources making enzymes for a sugar that's not even there. Okay? So each operon stays repressed, right? The repressor protein stays bound to the operator sequence and stops any transcription from happening unless the target sugar is actually there, okay? So if the target sugar, the ribose or lactose, is there, then that is going to take the regulatory protein, right, the RC protein or the lac repressor, away from the operator sequence of the operon, and then in that case, you would actually express that operon to digest the sugar that's present. So there are a few things I want you to notice about these operons. So first, notice that even when there's ribose around and the RC protein is no longer bound to the operator sequence, the other end of this RC protein stays attached to the initiator region of the ribose operon. So even when ribose has come along and bound to the RC protein and therefore helped it to detach from the operator sequence, so it's no longer looping the DNA, no longer blocking things, it stays attached to the the initiator region all the time. This is because the RC protein actually does a second function. So when arabinose is present, so the RC protein and arabinose complex here, it actually stays bound to the RI region, and from there helps RNA polymerase to bind. So this helps to initiate even more transcription when there's arabinose around. Okay, so it's kind of a dual function protein. So the reason why we don't call the RC protein a repressor is because it doesn't just repress. As much as one end of it can repress transcription when it's bound to the operator sequence, when it's not bound to the operator sequence and it's bound to arabinose instead, it has kind of a positive effect on transcription, right? So activator is a repressor when arabinose is absent, and an activator when arabinose is present. Okay, so we just call it the RSC protein, the control protein, that can either turn off or turn on, depending on whether arabinose is absent or present. Okay, so that's one thing to notice. Now, next, I want you to think about the effectors. This is just a review of allosteric effectors, meaning that the environmental kind of compounds that will bind to regulatory proteins to change their affinity to DNA and hence affect the regulation of these operons. I want you to think about what the allosteric effectors are for the lac and arabinose operons, remembering that each of these has two regulatory proteins that will bind to them. Okay, so here are your possible answers. Please pause the video and make your choice. All right, welcome back. So the answer is D. So both of them make use of the same activator protein, which is CAP, and therefore both of those CAPs need cyclic AMP as its effector molecule to activate them, right? But for their other regulatory protein for negative control, there is arabinose acts as the effector molecule for the RC protein, whereas lactose acts as the effector molecule for the repressor protein. Okay? All right, so hopefully that makes sense to you. If not, please do review these parts of the lecture first so you fully understand both the lactose and arabinose operons so you don't get them mixed them up with the next step, because next we're going to look at other kinds of operons that are not about digesting sugars, right? So note that when it comes to operons, this whole idea that you use a single promoter region to control the expression of a whole bunch of genes, a bunch of structural genes at the same time, that's pretty common in prokaryotes, but you don't really see them in eukaryotes very often. So when we're talking about operons, we're always talking about prokaryotes. So the same goes for the next type of operon we're going to talk about right now. So let's say that you have a biosynthetic pathway that you need in order to make something useful like an amino acid. So we talked about biosynthetic pathways quite a bit in terms of gene interactions before. So the enzymes that catalyze each of these reactions that make up the biosynthetic pathway, they often have their genes organized into organized as structural genes of a single operon, right? And kind of surprisingly, if you look at the operon for, say, that produces, say, all these enzymes for the biosynthetic pathway that makes tryptophan, right? They actually have their structural genes transcribed in the same order that the enzymes need to be used in the biosynthetic pathways. So notice that we're going to transcribe from left to right here. And the first couple of genes, structural genes, will be transcribed and translated to make the first couple of enzymes in the biosynthetic pathway. Then the third gene that gets transcribed is the third enzyme for this reaction, and the fourth for the next enzymes. So it's kind of in the right order. Now, this could just be a coincidence, but technically speaking, if you think about it, that actually makes things more efficient because that in E. coli cells, as you transcribe these structural genes, you can immediately start to translate them because there's no nuclear envelope separating transcription from translation. So it makes sense that as you transcribe this and you immediately translate the first structural gene, then you get the first enzyme for the first step of this reaction sooner. So this reaction can start happening even while you're still transcribing and translating the next genes, right? And so therefore the very next enzyme you start translating and seeing and being active is the next one needed in the biosynthetic pathway. So if you think about in terms of natural selection, an individual that has these genes in the same order as the biosynthetic pathway, they're going to start the early reactions and kind of get the enzymes they need sooner if they're in the correct order than if they're in the wrong order. All right, so the natural selection should favor those individuals with operons with structural genes in the same order as the biosynthetic pathway. That would make them a little more successful and quicker at producing tryptophan than those that have them in the wrong order. All right, so just an interesting thing to think about. So we're going to focus in on this tryptophan operon, the one that encodes all these enzymes for making tryptophan, right? And notice again that the enzymes needed to make this amino acid are encoded in order in this operon. All right. So notice that thinking back, we talked about different oxytrophic mutations that cause a bacterial or yeast cell, for example, to not be able to make its own amino acid like methionine, tryptophan, that kind of thing. So what we're actually talking about there is not just a single gene, but actually we're talking about mutations in operons, in the tryptophan operon, right? So that was actually an operon and not just a single gene. Okay, it's just an interesting thing to link back to. So let's look at how the tryptophan operon is regulated. So we're now looking at an operon not for digesting and using a particular sugar for energy, but for the synthesizing of nutrients. All right. So the regulation is a bit different from the lac and arabinose operons because the expression of these sugar catabolizing operons, the ones that digest lactose and digest arabinose, these happen to be induced by the presence of these sugars. So when lactose is around, that causes the lac operon to be expressed so you can digest that sugar. But if you're looking at an operon that's meant to produce a nutrient like tryptophan, then expression should be activated only when there's not enough tryptophan in the cell. All right, so it's kind of the opposite in that we have chemicals turning on expression when you know that you need these enzymes to break down what turned it on. Whereas in this case, when the product is tryptophan, then it's only when there's not enough tryptophan around that should turn on the operon so we can make more, right? So when there's lots of tryptophan around, that should actually repress expression of the operon. So you'll see this reflected in the structure and function of the tryptophan operon. All right, so let's take a closer look at how the tryptophan operon is regulated. Okay, so there's two different mechanisms by which the tryptophan operon is regulated. So the first one is going to be somewhat familiar, but you'll see that it's flipped around. So when the cell has enough tryptophan around and doesn't need to make any more, then it makes sense that you want a repressor protein to come along and block this operon so we're not going to create more than we need. Okay, so for this TRP repressor, right, so you have the separate gene here called TRPR. That's when it's transcribed and translated, produces the TRP repressor protein. Okay, and when the TRP repressor protein binds to some of the available tryptophan floating around the cell, right, then that tryptophan acts as a co-repressor and changes the shape of the TRP repressor, making it kind of have higher affinity to the operator sequence. So the repressor protein binds to the operator sequence when there's tryptophan around, and that is going to block transcription. Okay, so this is negative regulation again. Negative regulation because the binding of the regulatory protein blocks gene expression or operon expression. So similar to what we saw for the sugar operons, except that the effector molecule, right, the tryptophan, when it binds to the repressor protein, it actually makes it bind stronger to the operator sequence instead of making it come off of the operator sequence. So opposite direction. When there's tryptophan around, let's shut this down. We don't need to make any more. Okay, so we're basically increasing its binding to the operator sequence instead of decreasing it. So again, this makes sense. When you have enough tryptophan, you should block transcription of the genes for making even more tryptophan than you need. Okay, so the lac repressor, remember, it was an example of an inducible system because you can induce transcription by adding lactose. So adding lactose gets rid of the repressor and induces expression of the operon. So considering that, what might you call the trip repressor then? What kind of control would that be? So it's not an inducible system because adding trip doesn't induce expression of the operon. So what might you call this? So pause the video and make your best guess. Okay, welcome back. So I would call this a repressible system, right? The kind of opposite of inducible would be repressible, in that something in the environment, the tryptophan, can cause repression of the gene expression, right? When there's lots of tryptophan around, it's going to bind to the repressor and cause it to repress expression even more. Okay? So here's another a clicker question for you to think about. So let's say there's a mutation in this TRPR gene, right? The one that's transcribed and translated to make the TRP repressor. Let's say there's a mutation in this gene causing the repressor to be kind of messed up in such a way that it can no longer bind to tryptophan, right? So there's a mutation in the tryptophan binding site, in the gene in such a way that the tryptophan binding sites of this repressor protein no longer bind to tryptophan. What do you think will happen? So pause the video and come up with your answer. All right, so hopefully you came up with E. It's not an easy question, so if you came up with E, that's very good. All right, so if your repressor protein can't bind to tryptophan, that means tryptophan cannot kind of turn on the DNA binding site. So if that's the case, if tryptophan can never bind, then the repressor can never bind. And if you can't bind a repressor, that means you're going to get constitutive transcription of the operon because you're no longer blocking it with the repressor protein. Okay, so hopefully that makes sense. If there's any questions, feel free to give me a call anytime or to contact your TAs. So earlier I said that there were two different mechanisms for regulating this operon, right? One of which was the repressor protein, which will bind on whenever there's already plenty of tryptophan around so that we no longer express an operon that we don't need for now. The other mechanism is attenuation, and this is quite a different one now. So attenuation is when you start off transcription, but then you stop it early, right? You terminate transcription early at this attenuator sequence here. And this happens when there's lots of tryptophan around. So this is another way to stop expression of this operon when there's enough tryptophan around already. Okay, so that's a way to save on the time and energy it's going to take to finish transcribing all those structural genes, of which there's a lot more DNA to transcribe an RNA later on. And also just so you're not using all these resources to make the enzymes and make the tryptophan that your body doesn't need. Okay, so to understand how this happens, how is it that the presence of tryptophan in the cell can actually cause transcription to end early, let's take a look at how transcription normally ends to begin with when you get to, say, the end of the gene. All right, so here's a quick review of something you've seen before. So how does termination of transcription happen? How do we stop transcription? So normally, transcription ends when the RNA polymerase has kind of finished transcribing the entire coding sequence of the gene and then some, and it reaches the termination sequence in the gene. And so termination sequence being a sequence of around 40 base pairs at the end of the gene that helps to stop transcription. So there's two important regions of this termination sequence, one of which is this really GC-rich region that once it's transcribed in RNA can form this little hairpin loop. And then right after that, there's a bunch of A nucleotides on the template strand, which gets base paired with a bunch of U nucleotides on the RNA. Okay, so if you're looking at the DNA, you see a bunch of G's and C's, a little bit of stuff, a bunch of G's and C's, and then a whole bunch of A's after that. Okay, so the intrinsic mechanism for termination, which is what's relevant here, so there's many different ways of mechanisms for terminating transcription, but for this one, what happens is, so this DNA gets transcribed into RNA, and as this RNA is transcribed, right, since these two yellow segments here can base pair with each other, that's what they do, right? They base pair together to form this kind of hairpin shaped loop in the mRNA being transcribed. You see here as well. DNA is opened up, you're transcribing onto RNA, and as you transcribe the termination sequence, the mRNA itself forms this hairpin loop, and then it hits a bunch of A nucleotides on the template strand and tries to add a bunch of U's there. Okay, so once it's transcribed to RNA, the GC-rich regions base pairs with itself to form the hairpin loop, and then RNA polymerase proceeds to add a bunch of U nucleotides to match those A nucleotides in the template strand's termination sequence. Now, these AU base pairs, having only two hydrogen bonds between each complementary base pair, they're relatively unstable. So usually when it reaches a bunch of AU pairings, RNA polymerase is going to backtrack and try to find a stable GC pairing to stabilize itself before it continues transcribing further down the DNA. So because this hairpin loop has all these triple bonds holding it together, RNA polymerase is basically stuck right around here, right? It hit a bunch of UA kind of pairings and it wants to go backwards, but it can't because the mRNA behind it is already linked to itself, forming this loop. So since RNA polymerase can't move forward because of the AU pairings making it unstable, it can't move backward to find a GC pairing because the very next GC pairing is already paired with other parts of the mRNA strand, then RNA polymerase just can't go forward, can't go backward. After a while, it just gives up and releases the newly fully formed RNA sequence as well as the DNA template, which is allowed to coil back into its double helix and go back into rest. All right, so that's how a termination sequence, how intrinsic mechanism for termination works at the end of a gene normally. So how does it relate to tripoperons then? So basically, let's say that transcription is happening, right? The RNA polymerase starts at the promoter region and needs to transcribe through this leader sequence before it gets to the structural genes. Okay, so it's a really long leader sequence that's transcribed onto RNA. So this actually starts the promoter regions here. We start transcribing at the beginning of the leader sequence, and normally, if it's not being attenuated, the RNA polymerase will finish going across the entire operon, transcribing not just the leader sequence, but all of the structural genes all the way to the end, stop at the termination sequence at the end. Okay? But the leader sequence also has this early termination sequence called the attenuator. Okay? So there's a possibility of terminating transcription right here before it goes through all the structural genes and gets to the normal termination sequence here. Okay, so for the attenuator, when it's transcribed, the mRNA ends up forming a loop, right? And it forms the loop just before a bunch of A nucleotides on the DNA. So those UA base pairs makes things unstable. Okay, so here's how the decision's made and affected by tryptophan. So when there's lots of tryptophan around and you don't need to make any more, then that hairpin loop will persist within this attenuator sequence. Right, so once this attenuator sequence is sequenced onto RNA, and this loop remains unbroken, then transcription ends up ending early. Right, this acts as a termination sequence that ends transcription before you get to the structural genes. But if there is very little tryptophan around and you need to make some more, then something comes along and opens up this hairpin loop. Okay, as this mRNA is being transcribed from the attenuator. Right, so by opening up the hairpin loop, then the RNA polymerase is able to backtrack, find its GC pairing, stabilize, and continue moving forward and transcribing the rest of the structural genes. All right, so those are the two situations. If there's high tryptophan, just stop transcription here, don't waste our time and energy. If it's low tryptophan and you want to make some more, then get past this attenuator sequence, do not terminate there, continue transcribing the whole polycistronic mRNA and allow for translation to occur. Okay, so here's a challenge question for you to think about. Okay, so in this attenuator sequence here, you're basically transcribing a termination sequence, right? So this hairpin loop causes transcription to end early. Okay, normal. If there's lots of tryptophan around already, all right. So this is what causes the RNA to be attenuated to be shorter than expected and cause it to not contain any structural genes. The question for you to think about, this is a really challenging question, is what the heck can actually open up that hairpin loop, right? What is it that's opening that loop to allow for transcription to get past this early termination sequence and still continue to transcribe the structural genes? So it's a hard question, but see if you can figure it out with whatever logical way that you can. All right, so pause the video now and think about it. All right, welcome back. So before I reveal the answer, I'm going to give you a little hint, okay? So this process only occurs in prokaryotes, okay? So this way of opening up this hairpin loop in order to let the RNA polymerase backtrack, find a GC pairing, and keep moving and get past the attenuator to the structural genes, this can only work in prokaryotes. All right, so pause the video again and see if you can figure out your final answer. Okay, welcome back. So if you chose B, even if it was a wild guess, great job. You're a genius, because I don't think I could have figured that out my first time either. All right, let's talk about each possibility here. So for RNA polymerase, that can't really break the hairpin loop, right? Remember, the whole reason why this actually works as a intrinsic mechanism for termination is that RNA polymerase cannot break the hairpin loop. That's what makes it stuck here between those AU pairings and the hairpin loop. It can't break to find the GC pairings, and that's why termination will occur. So RNA polymerase cannot break the hairpin loop. It's stuck downstream with these AU pairings trying to back up and failing to do so. Now, in ribosomes, remember, in prokaryotes, because there's no nuclear envelope separating this transcription that's happening on the DNA from the translation that's going to happen in the cytoplasm with the ribosomes translating the mRNA here, then translation can happen during transcription. So in eukaryotes, since ribosomes are not found in the nucleus where transcription is happening, ribosomes can't come in to help. But in prokaryotes, since ribosomes and the DNA are together in the cytoplasm, a ribosome can come along and start translating this RNA as it was being transcribed, and as it reaches the hairpin loop, can actually prop open that loop, therefore allowing RNA polymerase to backtrack into a GC pairing and then continue transcription from there, past the attenuator through the structural genes, and finish transcribing the entire polycistronic mRNA from this entire coding sequence of the operon. Okay, so ribosome is the thing that's coming along this mRNA molecule and that can open up this hairpin loop long enough for RNA polymerase to backtrack and then move through the rest of the operon. Let's finish considering the rest of these options. So the TRP repressor protein, it's involved, but it's involved way back here, and it's binding to the DNA molecule. It doesn't really have the ability to bind to or affect the RNA molecule being transcribed way down here. Okay. Also, we don't really want the repressor protein to be binding around here because we're talking about a situation where we want to open the hairpin loop so that we can actually finish transcription and express these structural genes. So repressor proteins here to stop transcription, not allow transcription to continue. Similarly, tryptophan amino acids aren't going to bind to mRNA. Amino acids by themselves don't have the ability to break apart hydrogen bonds. And also, we don't want tryptophan to be what increases full transcription of the tryptophan operon, because we kind of want the opposite thing. We want it so that when tryptophan's around, we actually stop expression instead of increase expression. Okay, so logic points in the wrong direction for these two answers. All right, but if you chose B, great job. If not, hopefully you understand that that was the only other physical structure that can come along this mRNA and open up the hairpin loop, because the downstream, there's RNA polymerase stuck there and about to give up. And so in this end of the mRNA, ribosomes can kind of break it open. Okay, I took a little more time to explain that than I normally get to do in the lecture, so that's kind of a benefit of these videos, is I can take as much time to explain things as I like, and hopefully the result is that you'll actually understand it better than if we were together in the lecture hall. So, moving on. How does that work? How does a ribosome actually disrupt that loop to allow for transcription to finish when there's not enough tryptophan around, when there's a low concentration of tryptophan? That's what I mean by these square brackets. If you remember your chemistry symbology, that indicates concentration of what's inside, so concentration of tryptophan. So when concentration of tryptophan is low, let's look at what happens. So normally speaking, right, as you open up the DNA and you're transcribing your way through the tryptophan operon, and you're going through the liquid leader sequence, and you've gotten to this attenuator sequence, and you've transcribe that attenuator sequence onto mRNA, okay? There's these 4 regions in the attenuator sequence, right, that can actually base pair with each other. So it's actually not just a single loop, there's actually kind of 2 loops that get formed, okay? So as you transcribe the DNA, which is the blue here, onto RNA, which is the pink here, okay, notice that the RNA starts to base pair with itself, right? So we have a base pairing of this region with this region and this region with this region, okay? So if there's nothing blocking, if the ribosome's not here yet, regions 1 and 2 bind together, regions 3 and 4 bind together. So all of these segments that want to bind have something to bind with. So that's great. So regions 1 and 2 are base paired. Regions 3 and 4 can also base pair. And region 3 and 4, that's the important part because that is the loop that comes right before that string of U nucleotides that matches the string of A nucleotides on the template strand. Right? So this is the loop, then the AU pairings. This is where the action is in terms of are we going to stop transcription here or not. Okay? So as long as regions 3 and 4 stay stuck together, then RNA polymerase is going to get stuck here, not able to move forward past the AU pairings yet because it needs to backtrack, but it can't backtrack because this loop is blocking all the GC pairings. Okay? But so what happens when concentration of tryptophan is low that would actually allow transcription to continue and not terminate at this attenuation sequence? Well, notice that the ribosome has started doing translation at this start codon here and is basically moving along and translating this leader sequence and building a polypeptide. Not a useful polypeptide, but it's still moving along, just seeing what sequence is there and adding amino acids, and eventually gets to these two codons that code for tryptophan. All right, so it's basically asking the cell to bring two tryptophan amino acids in to continue allowing translation to happen. Okay, now if the cell is very low in tryptophan, then the ribosome is going to be stuck waiting here quite a long time, waiting for tryptophan to be brought to the ribosome. Okay, but if it's really low in concentration of tryptophan, it's going to be stuck here for a while. And while it's stuck here, it's blocking region 1, right? It's stopping region 1 from binding to region 2. So with region 1 blocked, region 2 is going to bind to the next closest complementary sequence, which is region 3. Right, so these two are also complementary, so they bind to each other. So regions 2 and 3 are binding instead. So therefore, with 3 occupied with region 2, region 4, notice that the 3 and 4 loop is now opened up. Right? With this opened up, RNA polymerase can now backtrack into region 4, find a GC pairing, and then happily move forward and continue transcription from there without having been terminated early. Okay? So the way we open up regions 3 and 4 is a little bit indirect, right? The ribosome translates along and ends up waiting for some tryptophan to arrive here for a while. And while it's there, it's going to disrupt the pairings between 1 and 2. Therefore, 2 and 3 need to bind, and then 3 and 4 are no longer bound together. By opening up the 3 and 4 loop, then RNA polymerase can backtrack and continue backtrack to find the GC pairing and then continues transcribing the rest of the operon, including all of the structural genes. Okay, so this describes kind of all the steps of what's happening when concentration of tryptophan is low and when the ribosome stops at those two tryptophan codons. Okay, what happens when tryptophan concentration is high? Well, if tryptophan concentration is high, then the ribosome's not getting stuck in the middle of region 1 anymore. It's not getting stuck at these two tryptophan codons here. So instead, it continues on and then ends up stopping at the stop codon, which is kind of near the end of region 1 instead. Okay, so at this position, the ribosome now being stuck quite a bit further, it's already added its two tryptophan amino acids and it's stuck over here, further down. It's going to block both regions 1 and 2, right? So neither of them combine with anything. So all that leaves open and looking for binding is regions 3 and 4, so they get to stay together now, right? So with regions 3 and 4 staying together, RNA polymerase can no longer go forward, it can no longer go backward because this hairpin loop is intact once again. So as a result, 3 and 4 stay together, RNA polymerase can't go forward, can't go backward, so it detaches and termination occurs, right? Attenuation occurs. We stop transcription early before we even get to the structural genes. Okay, so this does confuse a lot of people, so do review these last 2 slides again and try to make sense of it. If it still doesn't make sense, then give me a call or try looking at some videos online. Do put the work in to make sure this makes sense to you. Okay, a bit of a review question next to help you think this through one more time. So, what are the things that happen when tryptophan concentration is low? Here are your choices. Go ahead. All right, welcome back. So the answer is E, all of the above. All right. So just remember, both the repressor and the attenuation mechanisms, they're active all the time. And so um, when tryptophan is abundant, then it's going to be binding to the repressor protein and helping it to bind to the operator sequence, so it's blocking transcription that way. And also, if there is some transcription still happening, then it gets attenuated early before you end up transcribing and translating the structural genes as well. All right. So what's the overall effect of these two regulatory mechanisms? So when tryptophan is low, then um, the repressor protein doesn't have tryptophan attached to it most of the time, and therefore it doesn't bind very well to the operator sequence, so it's no longer blocking things. So the net effect of that is that the repressor protein is now removed from the operator sequence because without the tryptophan present, it just doesn't bind to the DNA very tightly. And that allows for transcription to happen a lot more because it's not being blocked. So you get about 70 times more transcription with the repressor protein removed than if it was still attached to the operator sequence. You also get less attenuation as well, right? So because of the attenuation kind of mechanism we talked about earlier, with low concentration of tryptophan, the repressor protein gets stuck at the tryptophan codons, and therefore there's less attenuation and more complete transcription of the operon, and that gives you about 10 times more complete transcription as before. So together, when tryptophan concentrations are low, you'll see about a 700 times higher amount of operon expression, tryptophan expression, compared to when there's lots of tryptophan around. Okay, so notice that in reality, there's no, it's not exactly like when the repressor is bound, it's not 100% blocked. It's kind of like it binds most of the time, but sometimes it's off, and sometimes a bit of transcription happening, and most of the time it gets attenuated, but sometimes it can complete, right? So there's quite a bit of gray area there, but we are kind of just looking at overall, is it mostly fully being expressed or not being expressed? Okay, so if you have any questions, do feel free to again email or call me or your TAs. But one more thing I want to talk about. So attenuation of other amino acid pathways. So we talked about one operon, the tryptophan, for producing a nutrient, so the tryptophan amino acid. But what about other amino acid operons? Is there attenuation in those as well? And the answer is yes. So you can kind of see these are the key parts of the leader sequences for each of these operons where you would find the codons coding for the amino acid that the operon is meant to make. So for the trip operon, here are the two trip codons. For the phenylalanine codon, here are the phenylalanine codons. Histidine codon operon, here are the histidine codons. All right. So notice that it's a little bit different for each of these amino acid producing operons, in that for the trip operon, as long as you have a low enough concentration of tryptophan that there's not a lot of these tRNAs holding tryptophan available, then the ribosome is going to be stuck waiting within this region 1 for a while. So if region 1 can't bind to region 2, then region 2 binds to region 3, and then the region 3 and 4 loop is broken, and hence the RNA polymerase can backtrack, find the GC pairing, and keep transcribing the rest of the operon. And so basically, not having not having two tRNAs holding tryptophan readily available will prevent attenuation from happening and allow transcription of the whole operon, allow expression of the operon. So, and as a result, you'll end up making more tryptophan. Okay, so basically, not having two tryptophans available is a sign that you need to make some more, is the simple interpretation of that situation. So with that simple interpretation, look at what's happening with the phenylalanine operon, right? So if there are 7 phenylalanine tRNAs available, right? Or if there's less than 7 available, then the ribosome gets stuck and the phenylalanine operon gets fully transcribed and expressed to make more phenylalanine. All right. So histidine as well has 7 of those codons. All right. So why is there this difference? Well, if you think about it, right, certain amino acids might actually be needed at higher concentration in the cell overall than others. Like for example, let's say that in proteins, maybe phenylalanine and histidine amino acids are more often used in the formation of proteins than tryptophan, right? If that's the case, then the cell should maintain a high concentration of these two amino acids in the cell so that they have the amount needed whenever it's making proteins. So by having more of these amino acids here, you're basically saying that you would need, in general, like say more than 3 times as much phenylalanine in the cell than tryptophan, because at the same concentration, enough concentration to shut down the tryptophan operon early to attenuate the tryptophan one, phenylalanine says, no, that's still not enough. I want so much. I want like more than 3 times more than that around before I start attenuating, right? So hopefully that makes sense. So that's just helping you to look at the different operons, and it's just a really interesting thing to think that built into the leader sequence of each of these operons is basically a certain number of codons that basically sets up how much phenylalanine should be in the cell compared to the amount of tryptophan, right? Because the larger number of codons that are present here, then the higher the concentration of phenylalanine you're going to need in order to get past this leader sequence fast and allow for attenuation to stop making more phenylalanine. Okay, same thing goes for the histidine.
Right, so one last thing I want to do is to do a little quick comprehension quiz with you guys. So I don't know if you remember from earlier in the semester, but myself, Kathleen Fitzpatrick, who has been teaching BIS 202 for many, many more years than I have, and Jonathan Uriel, who is one of our favorite and best TAs, the three of us are working together to write a free online textbook, an open textbook for BIS 202 for SFU students and anyone across the world to use for free. Just because we wanted to do that. We're not being paid to do it or anything like that. But as part of this project, we wrote a prototype chapter for the non-Mendelian genetic stuff, which you looked at before midterm 1, and then now for the lac operon stuff, because it is a very difficult topic. So we wrote a chapter for this. So what I want to do is I want to test your comprehension now that you've finished both lac operon lectures and haven't looked at the free chapter yet. And then at the end of this quiz, I'll give you the link to the chapter so that this basically becomes your pre-looking at the module quiz, and we can compare it to how you do in the final based on how much you use the resource, and we can get a measure of how well the module works, how much you're learning from it. So this is your before you look at the free chapter, and then the final exam will be looking at how much you learned from this chapter afterwards based on how much you used it. All right, so what we'll do later on is we'll kind of compare your results from this quiz with your final exam results, as well as your response to an online survey and the bonus question on the final exam that tells us how much time you actually spent using the chapter, just as a way to find out kind of how useful the chapter is, and also to find ways to improve it as well, so we can learn from you guys, your feedback, in order to make the rest of the chapters for this textbook as useful as possible for future students. So we really appreciate your help with this, and this will be completely anonymous. It's also not necessary as well as an optional part of this course. Okay, so for this quiz, it's only 4 questions long. It's only going to take you literally 5 minutes, but please answer independently. So meaning, don't, if you're watching this video with some classmates, please answer the questions. Each of you enter it on your own in the lecture participation quiz in Canvas. But also, unlike your other clicker questions, please leave this video running so I can kind of time out how much time you spent on each quiz question, so it's consistent across all students. Right, so I'll give you 1 minute per question for the first 3 questions, and then 2 minutes for the last question because it's a little bit harder, so that's 5 minutes in total. Okay, so these questions are not for marks. If you get them wrong, that's totally fine. They're meant to be kind of hard exam level questions to be done right now at the end of the lecture before you've read anything else. And it's also worth doing even if you're not interested in this free textbook stuff at all, because it'll help you understand the material to be quizzed on it right now, having finished watching your lecture videos. Okay, so we're going to start the question now, so make sure you get comfortable and have your Canvas lecture participation quiz ready, because you'll have exactly 1 minute per for each of these first questions. And so remember, please leave the video running, and I'll tell you when there's 10 seconds left, 5 seconds, and then when to finish entering your answer so we can move on to the next question. So enter your answer whenever you're ready. Okay, so here comes your first question. You'll have 1 minute starting now. Please leave the video running. 10 more seconds. Please enter your answer soon. And 5, 4, 3, 2, 1, and stop. Okay, so get ready for your next question, please. And go ahead. And 10 more seconds. And 5, 4, 3, 2, 1, and time's up. I do apologize for the traffic sounds in the background. I'm charging my car near a highway, near a busy road right now. Okay, ready? Next question starting now. Seconds. 5, 4, 3, 2, 1, and time's up. And here's your last question. You'll have 2 minutes to do it. Ready? And we'll start. Still have 1 whole minute. So you're halfway through. And 10 seconds. 5, 4, 3, 2, 1, and time is up. All right, thank you so much for participating in this. So now that you've done this quiz and finished your lectures for the Lac Operon topic, here's the link to our chapter. This is also in your slides as well, so you can use that to click on it as well. So again, you don't have to use this free chapter if you don't want to, but know that we basically wrote this specifically with you in mind as the readers of this chapter, and it's made for you to use right now when you've finished watching your lectures and are wanting to kind of refresh and deepen your understanding of this material through reading and practice. And we've made it as concise as possible and as useful as possible for you. So I do encourage you to use it. I think it will be good for worth your time and good for your learning and performance in your exams. All right. So some of you who understand how research works will think about, oh, okay, maybe they're going to use the exact same questions before and after you use the chapter. But of course, I'm not going to do that. Having given you questions, I'm of course going to give you the correct answers after this slide. And therefore, I'm not going to give you the same questions on the exam. There'll be different questions on the exam. But a benefit of this having you guys take part in this is that you will get another bonus mark on your final exam, just like you did for midterm 1. So the very last question of part 2 for your final exam is going to be this question, which you can read in advance if you want, so you don't have to waste time during the final exam. But just like the previous question, is basically asking you how much time you spent reading the text part of this free chapter and how much time you spent answering the questions. So it could be like, 45 minutes, 20 minutes, something like that, right? Or you can just put 0, 0 if you didn't use the chapter at all. But like I said, you don't have to even answer the question if you don't want to. If you want to opt out, you can just write opt out into that kind of text box that you can enter your answer in, and you don't have to answer it all if you don't want to. You'll get the bonus marks as long as you write anything in that box. Okay, so again, I'm not going to look at these answers until after the semester ends. This will not affect your mark at all. This is just very useful feedback for us and data for us as we prepare this free textbook for everybody in the future. All right, so here are the answers to the remaining questions. So this is the answer to question 1, right? So the answer is yes to all of the above, because the repressor region of the lac operon, or repressor as a regulatory element, is unique in that it does code for a protein, unlike, say, the operator sequence or the promoter sequence, which are part of the promoter of the lac operon, right, which don't make their own proteins. It's also the only transregulatory element, since it's the only part that actually makes a protein. It's the only one that actually has a protein that can float between multiple lac operons and affect more than one operon. And also, because it is its own gene that makes its own protein, it's not regulated by the lac operon itself. Right? It's its own separate gene somewhere else in the genome that makes this protein, and the protein comes to the lac operon to control it. That's why each of these 3 statements are true. If I went to that really quickly just because I just want to finish this video before it drags any longer than it already has, but do feel free to ask me more about this in office hours. For this question, I know these 2 things are not very familiar to you, but this is the best answer. So this being a multiple choice question, it's part of the test to see how well you understand or remember this information. So it makes sense that the lac operon is an inducible system, because you don't want to turn it on all the time, wasting amino acids to make all these proteins when you don't even have lactose to digest, right? So you should only induce this lac operon system when there's actually some lactose around to be digested, so you're not wasting amino acids making enzymes that you don't need. As far as these are the answers, like glucose is not toxic even if there's lots of it. Although maybe to humans, if we have too much sugar, yes, we do get kind of obese. It's bad for our health. And the permease enzyme is not leaky. Generally speaking, transport membranes are very, very specific, so it's not going to let other things through if it's made for lactose only. For this question here, it's a matter of interpreting this genotype here, or this description of the lac operon. So you see that the promoter is working, so you can do transcription. The operator sequence is a constitutive expression one, which means it's never going to be blocked by the repressor protein, so you're going to constantly express all of the structural genes. But the Z one is mutated, so it doesn't produce working enzyme, and so it's only the Y structural gene that's expressed and produces permease. So permease is produced all the time, but not beta-galactosidase, in a situation where lactose is present and there's glucose absent. Okay, lactose, remember, the absence of glucose will mean the expression will be strong. The presence of lactose in this case doesn't actually matter because we have a constitutive expression operator sequence. Okay, and for this last one, it's a matter of interpreting each of these genotypes. So I'll go through really quickly. So notice working promoter, so you can do transcription. Normal operator sequence, which means that repressor proteins can bind to it, but with a super repressor being made, it's going to bind permanently to the operator sequence, hence blocking any transcription from happening. Therefore, you'll see no production of permease or beta-galactosidase in a cell with this genotype. Okay, this one here again, working promoter, working operator, looks like everything's a go. We have production of normal repressor protein, which in the presence of lactose will not be binding to the operator sequence, so we're not blocking expression of this operon, and the absence of glucose means we're going to have strong expression, but the problem is what we're expressing are 2 mutated versions of these structural genes such that we're not making any active beta-galactosidase or permease because these structural genes themselves are mutated. And this last one here, the promoter sequence is mutated such that the RNA polymerase cannot bind, and therefore you'll get no expression. So therefore all of these are good answers. Okay, so that's pretty much it for this lecture. Sorry for all the traffic noise again. But yeah, so before I go, I'm just going to leave you with a quick silly video of Katie dancing around, because sometimes when you hear a good song, you just gotta dance. All right, enjoy everybody. Thanks again for taking part in this, and I'll talk to you soon. Okay, bye.